सैल्मोनेला। सैल्मोनेला हम स्टार्ट करेंगे सैल्मोनेला जीनस से। सैल्मोनेला जीनस इज कॉज लाइक एंट्रिक फीवर, गैस्ट्रोइंटेस्टाइटिस, सेप्टोसीमिया with or without focal suppression, and the carrier state. So like most important genus in this we have Salmonella typhi, and this Salmonella typhi cause typhoid fever. Then we have genus Salmonella. This Salmonella is divided into two groups. We call as typhoid Salmonella and non-typhoid Salmonella. Why we call this typhoid salmonella? Because they are the exclusive primary human parasites and they cause like enteric fever groups. So they include typhoid and a paratyphoid bacilli. We'll discuss it later on of typhoid and paratyphoid. Then we have non-typhoid salmonella, which cause like food poisoning groups. They cause animal parasite which can infect human, and basically they cause gastroenteritis, septosemia, and the local infection in the humans. Now we discuss the morphology. As they are Salmonella is gram-negative bacilli. They are motile with petricate flagella. So they have flagella all around the body. We have an exception in this. We call as like Salmonella gallinarum and Salmonella polorum. So they are non-motile. So basically they are exception, and please remember this. This can be asked in your MCQ. And then we have they are non-capsulated, non-sporing. They may have fimbriae, or they are aerobic and facultative anaerobe. So now we are moving to the culture property of this. So then we have the culture, the Salmonella is aerobic and the facultative anaerobe. So we discuss it, what the different types of culture they grow on the media. So we have MacConkey agars. So MacConkey agar, they does not have a lactose. They have non-lactose fermenting. So what they happen, generally they have the pale colonies can be seen, or we can say the colorless colonies can be seen in this MacConkey agar. Then we have deoxycholate citrate agar, we also call as DCA. They produce like black-headed colonies. So why they produce black-headed colonies? Generally they produce due to the production of H2S. So, and we have also xylose lysine deoxycholate, we call as XLD agar, and they also produce black-headed colonies due to production of H2S. Then we have Wilson and Blair's bismuth. Why we call as Wilson and Blair bismuth, and they generally does not have a specific role. They have like cause Salmonella to produce like colonies with a jet black with a metallic sheen. So this can be seen in the agar colonies. Then we have selenite and F broth. Or we can say that we have also a tetrathionate broth. They use in enrichment of this media. Then we have biochemical properties. So in biochemical properties, as we discussed, that Salmonella ferment glucose, mannitol and maltose. So they form acid as well as gas. So in this we have also exception which is called as Salmonella typhi. They does not produce gas and their behavior is like energenic. Why we call this energenic? Because it does not undergo any type of fermentation. They does not ferment is like sucrose, lactose and salicin. So they do not produce indole and they do not hydrolyze urea. So this is very important question. Which are the exception in the Salmonella typhi that do not produce gas and they do not hydrolyze urea. So we have Salmonella typhi. Please remember this. Now we discuss the antigenic structure of this Salmonella. So we have like four antigenic structure. We have flagellar antigen that we call as H antigen. We have somatic antigen that is O antigen. We have surface antigen we call as VI surface antigen, and then we have fimbriae antigen that is we call as F antigen. This is very important for our antigenic structure. Please remember this. And why we call as H antigen? Because they are present on the flagella. They are heat liable proteins. They are strongly immunogenic, and on the basis we have like if antibodies formation occur in the body, the rapidly secretion of this antibody will be high in the high titers following like injection or immunization. And we have like O antigen. As a O antigen is a somatic antigen, and in this we have why we call as somatic antigen? Because they have phospholipid protein polysaccharide complex, and they have like who discovered this? Discovered by the bovine antigen, bovine. So we call as bovine antigen because it's first shown by the bovine. Then we have the antibody to O antigen is cross reactive. If there is cross reactive, then the H antigen is more reliable as compared to the O antigen, because the H antigen has high capacity of strongly immunogenic as compared to the O antigen. Now we discuss as a VI antigen. Now this VI antigen is a surface polysaccharide antigen that's enveloping the O antigen. So we call as which antigen is enveloped by the VI antigen. So we call as it is enveloped by the O antigen. It is enveloped by the VI antigen to the O antigen. So we had this O antigen. In this VI antigen, we have analogous to the K antigen of coliformis. Then we call as what is the important function of this VI antigen. This VI antigen is a polysaccharide. As we discussed, they act as a virulence factor, like they inhibit the phagocytosis and the release complement activation system, and this will cause bacterial lysis by alternate pathway. And the second is called they also cause peroxidase mediate killing. So they cause killing by peroxidase medium. Now we discuss the pathogenicity. The Salmonella are strict parasite for animals and humans. We discussed it earlier also. So we have like S. typhi, S. typhi A, S. typhi V. They are confined to the humans. Then we have S. typhi murium, which have wide variety of range, which affects like animals. We have like they affects also birds, humans. If we talk about the clinical syndrome of this salmonella, we have like they cause enteric fever, gastroenteritis and food poisoning, septicemia with or without local supportive lesion. Then we discuss about like the how this pathogenicity occur of this enteric fever. First of all, this typhoid fever is caused by Salmonella typhi, and the paratyphoid fever is caused by S. paratyphi, which is of A, B, and C. And then the infection occur by ingestion. This after the ingestion, the bacilli will reach the gut and they will attach to the microvilli of ileal mucosa. Now they will attach to the ileal mucosa. What will happen? They penetrate the lamina propria. They will penetrate the lamina propria and the submucosa, and this will reach to like phagocyzed by macrophage. Now here they resist the killing. How they will resist the killing? They resist the killing by modify the lipopolysaccharide. They will change it. They will change the lipopolysaccharide of them, and they inhibit the phagolysosome formation. So they will resist the killing. Now if they will not kill, they will reach into the lymphatic system. Now they are reaching to the enter into the lymphatic system, where they will multiply, and through that thoracic duct they will enter into the bloodstream. After entering into the bloodstream, they will cause bacteremia. Please remember this. They will cause bacteremia, and after bacteremia they will enter into the various organs like liver, kidney, gallbladder, spleen and the bone marrow, where they further multiply. Now, if we talk about the clinical cause, we have most common age group of this will be of 5 to 20 years, and the incubation period will be like 7 to 14 days or 1 to 2 weeks. We have like onset symptom will be occur after it enteric will start. The gradual with headache will occur, anorexia can occur, the coating tongue and abdominal discomfort can be occur. This will lead to like constipation or frequent diarrhea. Then we talk about the typical feature. They will be like step ladder pyrexia. The pyrexia will be, the fever will be step like. They will be first, it will be increased like 1 to 2 to 3 days. So this fever will be increases day by day. Then in this bradycardia will occur, toxemia will also occur, and what can we see? The presence of soft palpable spleen can be seen, and hepatomegaly can be seen. Then what are the complications that include in it? The complications that include like intestinal perforation, hemorrhage, we have like circulatory collapse, we have like other complication which can be seen, which is more renowned, that is bronchitis, bronchopneumonia can be seen, some like psychosis, deafness, meningitis can be seen. Then we have other complication that can be seen, the arthritis, peritonitis, abscess, hemolytic anemia, thrombosis. This other complication can be seen. This is whole about the pathogenesis.